Okay, so thanks again. And we now move on to our last speaker of the section and of the day. It's my great pleasure to introduce my actually former colleague, Chen. And he is now the current chair of the Department of Surgery, and he is a professor of surgery, biomedical engineering, and he's now in UAB. He moved from Wisconsin to the warm place. Here you go, Herb.
Thanks. Now the last thing I want to do is be the person standing in front of you in a cold beverage and great music tonight. So I'm going to try to make my talk brief. And I'm not talking about a cardiovascular topic. I'm actually talking about Notch signaling to maybe give you guys an idea of the potential application of Notch outside of cardiovascular. You probably know Notch's role as more of a stem cell role, but we're going to talk about how we're actually utilizing the knowledge of Notch to hopefully suppress neuroendocrine cancer. And this is work that I'm so lucky to be. I was a member of the biomedical engineering faculty at UW, and now I'm glad that Jay has extended the same honor to me here at UAB. So just for those of you who don't deal with neuroendocrine cancer, they are basically hormone-secreting neoplasms that develop throughout the body in almost every organ. And so it allows us a great opportunity to study a cancer that generally can be anywhere in the body. And although they're relatively rare, they're highly metastatic, that most patients with this disease present with metastases when they see us in clinic. And so what our group has been doing over the last 17 years is studying the role of basically we study 3 of the 4 Notch family members and their role in these cancers, and we've also looked at other type of cancers. And many of you will be familiar with the Notch signaling pathway as shown here. And I'm not going to walk through it, but basically we know that it is a membrane receptor, which when one of its ligand bindings results in downstream signals, which basically result in changes at the transcriptional level. Now, the role of Notch, as you know, was first described in Drosophila, but much work in our interests became when this paper published in Nature really revealed the role of Notch's role in the development of the pancreas, that basically if you look at the neuroendocrine cells in the pancreas, and on the far left, that's what a normal pancreas looks like in the mouse, and the dark cells are the neuroendocrine cells. And those are about the normal distribution of neuroendocrine cells. But if you overexpress Notch in mice, basically all the neuroendocrine cells disappeared. But if you deleted Notch, you had an overproliferation of neuroendocrine cells, which got us to thinking that Notch, in contrast to its role in other organs and other cancers, where it usually stimulates proliferation, in the neuroendocrine cells, it was actually shutting down proliferation. So our first hypothesis is Notch was really lost in neuroendocrine cancers and really functioned as a tumor suppressor. And first we want to confirm that by looking at tissues from patients with neuroendocrine cancers. And the neuroendocrine cancers are shown, medullary thyroid cancers are neuroendocrine cancer. And we looked at the presence of the active portion of Notch1, the cleaved NICD. You could see it's absent or lost in neuroendocrine cancers. And so that was a universal observation we made in all neuroendocrine cancers tested to date. So our second question was, what if we restored the missing Notch in these neuroendocrine tumor cells? What would happen? And so we did this in a couple of different models, first an in vitro model and an in vivo model. But just what we did is we created an inducible active Notch1 and subsequently Notch3. And the data here shows that A is just a Western blot looking at these cells, which we genetically engineered to be doxycycline inducible. And if you look at B, the effect of what happened of a Notch induction, it completely suppresses proliferation of these cells in a dose-dependent manner. So if you look here, these are without doxycycline and absence of Notch, but as we increase the amount of Notch in a dose-dependent manner, in the highest dose of Notch, it completely stopped proliferation, and these are just the vector controls to say that it's not a result of doxycycline. So what we did is spent about 10 years showing that, in fact, Notch functioned as a tumor suppressor in all neuroendocrine cancer types, carcinoid, medullary thyroid cancer, and pheochromocytoma, and then basically decided to proceed with a strategy to activate Notch as a therapeutic target. And so how we decided to do that is we asked the question, are there any compounds that activate, that turn on Notch1 in neuroendocrine cancer cells? And so to do this, we decided to embark on compound screening through use of the small molecule screening facility. So what we happen to have handy is that one of the properties of Notch signaling, as probably many of you know, is that when the pathway gets activated and Notch goes under its 3 cleavages and the intracellular domain is released, it has to bind its binding partner, CBF1, to initiate these transcriptional events. So we were able then to create a method where we could measure Notch1 induction by this property of NICD binding to CBF1. And so what we did is we created or obtained a plasmid containing the CBF1 binding site fused to a luciferase, and we were able to then transiently transfect it into neuroendocrine cancer cells, creating a mechanism where we could use this to screen for any molecules which turned on or activated Notch signaling. And through our collaborators at Wisconsin, we went through the small molecule screening facility that Mike Hoffman runs. And we were able then to screen a whole bunch of the compounds that were present at the Wisconsin facility. And we started, first of all, with a known bioactive library, so drugs that were already known to used in humans and have established safety profile, because we were thinking immediately that we wanted to translate this. So actually, when the surgical resident, Scott Pinchot, did this in a matter of weeks and was able to screen a little over 7,000 compounds. And what we found, and this is, we found 27 compounds in this screening that passed the first and secondary hits, and those compounds are listed here on the x-axis, and on the y-axis were their luciferase activity. And 2 of the highest compounds shown here, one was valproic acid and 2 was resveratrol. And so we decided to proceed with valproic acid because it's a common drug used to treat epilepsy, confirmed by Western blot that indeed induced doses of valproic acid induce both the full length and the cleave notch in a dose-dependent manner. Confirmed in vitro, then showed this in an in vivo model of neuroendocrine cancer, that if we treated mice with neuroendocrine cancer cells embedded in them, we saw that in the control condition, the cells proliferated, but when treated with VPA, we could suppress the growth. So immediately with this data, we went to do a clinical trial. So what we did is take it from the bench to the bedside, and we decided to enroll, and this was a proof of concept clinical trial where we wanted to just show that we could induce Notch signaling in patients with metastatic neuroendocrine cancer. So what we did is we treated patients with valproic acid to the dose that we would use to treat epilepsy, because we found that those doses were effective to suppress tumor growth in mice. So we enrolled 7 patients, and to do this as a proof of principle, we obtained a biopsy of their tumors pre-treatment. So many of them got a liver biopsy. And then they had to agree to let us biopsy their tumor after treatment with valproic acid for 8 weeks. And again, our primary endpoint was look if we could turn on Notch signaling. And so this is briefly our results summarized, and this is basically taking the tumor tissue before and after valproic acid and looking at the RNA level for Notch1. And you can see it's not present before valproic acid treatment, and we had a high level of average induction, but a huge error bar, really representing that we were able to induce high levels in some patients, but in other patients it wasn't, we could not induce it much at all. And of course, although this is our primary endpoint to look at if we could functionally turn on Notch, we did look at tumor progression as measured by changes in their tumor markers, and you could see some patients continued their tumors proliferate, so it clearly had no effect. But interestingly, we did have some patients with reduction in their tumor growth. And because, remember, these patients had metastatic disease, to get on this trial, they had to fail all previous therapies, so they really had progressive disease. So it's not surprising that 6 of these 7 patients eventually died of progressive disease, but 1 patient actually lived, and it's actually this patient right here who had some tumor response. So we were encouraged by that, but also realized that we couldn't, it wouldn't work for everyone. And part of the reason was that in looking at this more. I'm not going to present data, but we found that virtually all HDAC inhibitors, which valproic acid is a class of, can induce Notch in neuroendocrine cancer tissues, but we needed relatively high doses to achieve this. So we had to come up with a better way to deliver these drugs to the tumor cells, and so we decided to embark in a collaboration with our colleagues in the biomedical engineering department to develop some nanoparticles that could do this. So what we did is decided to focus on utilizing nanoparticles which had somatostatin targeting receptors on them, and that's because somatostatin is a receptor that's expressed in virtually all neuroendocrine cancer types. And this is just an example of many neuroendocrine cancer tumors that stain positively for many of the subtypes, and this show is one of the somatostatin receptors. Virtually all, this is a profile of all cell lines, neuroendocrine cell lines that have these receptors on their surface. And so what we're able to do is show that these, the somatostatin receptors sit here on the membrane of these cells, and we can image them as shown here. We have animal models where we can actually image the presence of the receptors. And so we wanted to come up with a ligand which basically hit the receptors. One in clinical use is octreotide, but another one that is beginning to be in clinical use is called KE108, which has affinity or stronger affinity to all these receptors. And so we created a nanoparticle in collaboration with Sarah Gong, which basically encompasses all these different properties. A tumor targeting property with KE108, but also we packaged the drug inside. And then we also had a contrast agent to image them. And we could definitely show that if you looked at neuroendocrine tumor cells we developed the targeted nanoparticles and compared them to non-targeted, you can see that they robustly are taken up by the neuroendocrine cells as shown by the bright field, or shown by the red imaging by CY5 there. And so we're right now embarking to do that. We've shown efficacy in animal models, and we want to proceed with that, hopefully to get that to the bedside. And another mechanism we're also using is to, and this is a collaboration here with Margaret, where we're using antibody targeted methods to basically fuse anti-proliferative drugs to an antibody targeting somatostatin receptors. And Margaret and Renata in my laboratory have done great work with this, and the rationale behind using this concept is that using these ADCs, they're internalized by the cancer cells, and then the anti-cancer drug is released, resulting in cell death. We have some great models to test this, both in animal models here. But importantly, for neuroendocrine tumors, no one has established, neuroendocrine tumors don't grow outside of humans for some reason, so we've been limited in how we can investigate them. But through a collaboration with Joel Berry here, we're actually, for the first time reported, we can take human neuroendocrine cells and actually grow them in these chambers and these bioreactors, which allow us to do real-time clinical trials. So what we're hoping to do is utilizing these two methods to target neuroendocrine tumor cells and hopefully our Notch-inducing compounds, as we can do real clinical trials ex vivo in patients and decide what therapy is best for the patient and then deploy it for the patient. So I'd like to acknowledge our collaborators both at UAB elsewhere, our funding, and of course the people that do all the work in our lab, and just thank you for your time.
Thanks, Herbert, for the great talk. Questions? Yeah, John.
Nice talk.
Thanks.
I have a question for you. Antibody-directed targeting has been around for quite a while now.
Yes.
Is there anything that's made it into clinical trials or made it through clinical trials?
Oh, yes. So there are many ADCs that are used right now in treatment. And so that's why, I mean, we were focused on the nanoparticle, but that has not really successfully transitioned clinical use where ADCs have. And so I can't really take the credit. Margaret and Renata are the ones that really came up with the concept and pushed that through. And I think, so that's where we're putting most of our efforts right now.
So, if there's no other questions, let's thank the speakers again, and thank everybody for being awake and staying for the entire day. It's almost a full day. I mean, thank you all.